Two people can share the exact same DNA sequence, yet develop completely divergent health profiles. One identical twin might live a long, healthy life, while the other develops a severe chronic illness. If their genetic code is identical down to the molecular level, their biological outcomes should, in theory, match. The biological driver of this divergence is epigenetics, defined as heritable changes in gene expression. These changes occur without any alteration to the underlying DNA sequence. Genetics provides the baseline code, while epigenetics dictates the functional reality of the patient. Think of the human genome as a massive fixed reference book. Every cell in your body contains the exact same text. Epigenetics acts as the highlighters and bookmarks used by the reader. The highlighted sections represent genes that are turned on and actively expressed. The sections that are ignored fade away. These are genes that are turned off and silenced. The actual text of the book never changes. A liver cell and a skin cell have the identical manual, but they read completely different chapters. Activating different combinations of the exact same manual creates entirely different biological outcomes and health profiles. We can identify the first major regulatory pathway here, DNA methylation. Notice the pink arrow targeting the DNA strand to attach a specific chemical tag. This chart breaks down the precise biochemical reaction. A methyl group, a carbon atom bound to 3 hydrogens, is added to a cytosine base, converting deoxycytosine into 5-methylcytosine. The catalyst driving this addition is the enzyme DNA methyltransferase, or DNMT. This maps how methylation affects gene promoters at CpG islands. Unmethylated, the genetic machinery easily reads DNA, allowing active RNA transcription. However, hypermethylated islands attract repressors that block the machinery. As a result, transcription is completely abolished. This microscopic chemical tag becomes a clinical issue when it targets tumor suppressor genes. Tumor suppressor genes are responsible for halting unregulated cell growth. When methylation turns these specific genes off, the primary safeguard against cellular mutation is removed, increasing cancer risk. A simple chemical tag silences protective genes, driving a patient towards severe pathology like cancer. The second major mechanism controls gene expression through physical packaging, histone modification. This illustration shows how DNA is organized. To fit inside the microscopic nucleus, the DNA must be wrapped tightly around protein spools called histones. This diagram shows how chemical modifications alter structure. Adding acetyl groups spaces histones apart. Mnemonic, acetylation equals an active gene. Loose DNA allows access. Conversely, deacetylation removes groups, packing histones tightly. This creates condensed DNA that physically blocks machinery, keeping the gene silent. The physical spacing and structural access to the DNA ultimately determine whether a gene's instructions can be executed. The third regulatory pathway relies on non-coding RNA, specifically microRNA or mRNA. Standard RNA acts as a messenger to build proteins. mRNA does not. Instead, it acts as a regulator, intercepting messenger RNA and blocking gene expression before a protein can be formed. These mechanisms drive a spectrum of clinical diseases. Epigenetic silencing triggers neurological disorders like Alzheimer's and metabolic diseases like obesity and type 2 diabetes. Furthermore, errors in genomic imprinting cause specific developmental disorders like Prader-Willi and Angelman syndromes. Epigenetics acts as the regulatory bridge between the cellular environment and the expression of high-yield clinical pathologies. Unlike the underlying genetic code, epigenetic changes are chemically reversible. This flowchart illustrates the pathways of genetics and epigenetics. On the left, genetic disease is driven by irreversible structural damage to the DNA sequence. On the right, epigenetic disease is driven by the addition or removal of chemical tags. These epi-mutations are fully reversible. The addition or removal of these tags is driven by external factors. Diet, smoking, psychological stress, exercise routines, and environmental toxins all directly dictate which epigenetic marks are applied to the genome. Daily lifestyle choices and environmental exposures dynamically edit the biological expression of a patient's inherited DNA. Because these tags are reversible, they can be targeted pharmacologically through epigenetic therapy. There are currently 2 major classes of epigenetic drugs in use: DNA methylation inhibitors and histone deacetylase, or HDAC inhibitors. These drugs strip away the repressive chemical tags, causing the tightly condensed chromatin to uncoil and open up. The primary clinical application is in oncology, where they are used to reactivate the tumor suppressor genes that cancer cells have silenced. To synthesize this for board prep, review these final high-yield points. First, DNA is fixed, but gene expression is highly flexible. Second, gene activity is controlled by DNA methylation, histone acetylation, and non-coding RNA. Third, lifestyle and environment dictate these tags, driving the progression of cancer, aging, and chronic disease. The genetic code provides the biological blueprint, but the epigenetic landscape determines which parts of that blueprint are actually executed.